Okay, to get started. So where we're going to begin today is we're going to begin where we left off last time, of course, which was the story about Brown-Séquard. And Brown-Séquard was the idea of the hemisection of the spinal cord. And I want to walk through Brown-Séquard because if you understand Brown-Séquard, you really understand where the different sensory modalities are crossing over. That the pain and pressure and temperature are crossing over at the site where the sensory neurons are located, the neuronal cell bodies. And the proprioception and fine touch doesn't cross over until we get to the brainstem. And we'll walk through that and try to emphasize why the hemisection of spinal cord, Brown-Séquard syndrome, gives us some insights into the neuroanatomy. Then we're going to spend some time on what's going on at the site in the brain, the somatosensory cortex. The target for those third-order neurons, the third-order neurons are going to innervate neurons in the somatosensory cortex of the brain. And so what we're going to do is we're going to follow these third-order neurons and find onto neurons of the somatosensory cortex. And then what we're going to find is that there's a kind of map of the body plan on the somatosensory cortex. So there'll be regions of the face, which will be exaggerated in the somatosensory cortex. They'll receive sensory information from the facial areas, others of the hands. And we'll walk our way through this idea of mapping out the body plan on the surface of the brain. And then we're going to move from this region, which is going to be part of the post-central gyrus, a particular region of the brain, and we're going to jump over what's known as the central sulcus, or central fissure, and we're then going to find ourselves in the motor cortex, the precentral gyrus. And the precentral gyrus we'll find has the motor cortex, and the motor cortex is going to contain a series of cells called upper motor neurons. They have another name. The other name is they're called Bet cells. We don't like that name particularly anymore. This is a sort of an old-fashioned eponymous name, so it's better to call them upper motor neurons. They're unusual and they're enormous. Some of them are as much as 100 micrometers in diameter. They're among the largest cells in the body. And these Bet cells, the upper motor neurons, we're going to follow their path of the axons. We'll see the Bet cells form axons that enter something known as the internal capsule. Now we're going to simplify this. We're going to put only the upper motor neuron axons in the internal capsule. If you take a neuroanatomy course, you'll find there's sensory afferents in the internal capsule too, on their way to the postcentral gyrus. But we're going to emphasize the idea that in the internal capsule, we have the axons of these Bet cells, these upper motor neurons, and that they continue, they skirt around the thalamus, they find their way into the brainstem, and just about where the brainstem is going to meet the spinal cord, they cross over the midline. This crossing over has a name. It's called the decussation of the pyramids. Decussation is just a fancy Latin term meaning to cross over, and these upper motor neurons will have axons or processes that will cross over from one side to the other just before they enter the spinal cord. And then in the spinal cord, these axons will be in a bundle. That bundle will have a name. It's called the lateral corticospinal tract. Well, the lateral tells you where it is in the spinal cord, but the corticospinal term, this is what we talked about yesterday. That first syllable, cortico. Cortico tells you that the neuronal cell bodies are in the cortex. Spinal tells you the output regions are in the spinal cord. And we'll see that these upper motor neurons will pass all the way down the spinal cord until they reach the proper place during development, leave this lateral corticospinal tract, enter the ventral horn, and synapse on lower motor neurons. So the upper motor neurons have a direct synapse onto the lower motor neurons and drive those lower motor neurons. This is unusual for most animals. We find in primates, we don't see this really in other animals. It's mostly because primates have this ability to do what they call fractionated movements. We have very good movements of our hands. Also, there's very fine innervation, motor innervation in the muscles. But what we want to think about then is what the fate of the lower motor neuron is. We've talked about lower motor neurons quite a bit, but now we're going to see the upper motor neurons synapse directly onto the lower motor neurons. The lower motor neurons then leave the spinal cord and go out and innervate muscle. We've talked about this with the reflex arcs. And there's a definition here, though, called the motor unit. The motor unit is defined as the lower motor neuron and the group of muscle fibers it innervates. As we walk through this, what we'll see is that each lower motor neuron innervates several muscle fibers, but each muscle fiber receives input from only one lower motor neuron. What this means is that when an individual lower motor neuron fires an action potential or a series of action potentials, that all of the muscle fibers it innervates, it may be 20 or 50, it may be a few hundred, but all of those muscle fibers that are innervated by an individual lower motor neuron will simultaneously contract if things are working properly. That's the idea of the motor unit. That the lower motor neuron activates multiple muscle fibers. Then we'll talk a little bit about this intersection between the lower motor neuron and the neuromuscular synapse. And we've talked about this a little bit. We've talked a whole lot about synapses. So we can go through this fairly quickly in terms of the structure and function of the neuromuscular synapse. All the rules we've learned about synapses up to this point can be applied to the neuromuscular synapse. We're going to generate an excitatory postsynaptic potential by using acetylcholine, opening nicotinic acetylcholine receptors. Those are the nicotinic receptors that Langley found when he was walking along the muscle fiber using his brush full of nicotine. And then finally, we'll talk about the muscle fiber as a muscle cell. These muscle fibers are in fact individual cells. They're unusual in that they have many, many nuclei. They're said to be multinucleate. There may be several hundred nuclei in these muscle cells. And they're also unusual in that they have a complex system of membrane systems, and we'll talk about that. But let's start with this idea of Brown-Séquard. I'll try to do this fairly quickly, where there's the brainstem, there's the spinal cord. Out here, let's use the foot. We could use the hand, but let's use the foot, just like we did yesterday. And we're going to say, as though I'm facing the back of the board, we're going to say this is the right. And we'll say this is the left. And I'll remind you that in the quadriceps muscle, there was the input region of the stretch-activated system, these primary sensory afferents that have their neural cell body in the dorsal root ganglion. But these neurons, because they're going to be carrying information about proprioception, and fine touch. These neurons are going to enter the spinal cord and ascend the spinal cord on the same side until they reach the dorsal column nuclei. Just to abbreviate, this is the dorsal column nuclei. The second order neurons have their neuronal cell bodies, their input regions, the dorsal column nuclei. They cross over the midline and then these will then ascend the cord to the thalamus. And in the thalamus, the second order neurons have synapses onto third order neurons in these regions known as the thalamic relay nuclei. These third order neurons now extend processes, and they extend processes that end in the region known as the somatosensory cortex. That's part of the brain. So that's the pathway. That's the pathway we followed yesterday. And then we said that from the right side we could have a pain neuron. This could be pressure or temperature. Sends a process in. Also has this neural cell body in the dorsal root ganglion. Sends a process into the cord, and then almost immediately after entering the dorsal horn, that primary sensory afferent synapses onto a second order neuron, crosses over the midline, ascends the cord all the way to the thalamic relay nucleus and form synapses onto third-order neurons, which then extend processes to the somatosensory cortex. We can follow the same pattern from the left leg because we are, after all, bilaterally symmetrical. We can use an example of the stretch receptors in the quadriceps. We can see that this primary sensory afferent, again, has a neuronal cell body on the left side, enters the cord, ascends the cord on the same side, and then in the brainstem forms synapses onto the second-order neurons that cross over the midline and ascend the cord to the, this is going to again be the thalamic relay nuclei. And in the thalamic relay nuclei, those second-order neurons synapse onto third-order neurons. And the third-order neurons send processes to the somatosensory cortex. So let's now follow the pain neuron from this side. Here's the pain neuron down the tip of our foot. Has this neuronal cell body in the same segment, we'll say. Comes into the cord, immediately synapses onto the second-order neuron. Second-order neurons always cross the midline. Crosses the midline, ascends the cord all the way to the thalamic relay nuclei, where the third-order neuron sends a process now to the somatosensory cortex. That's the wiring diagram. Brown-Séquard is a case where there's been an accident, some sort of damage. And what we have done is we have cut the spinal cord right to the midline. Remind you that we have a midline in here. And the cord now is cut to the midline. The question to ask is what information is interrupted and what information gets through. So if we would touch this person with a light touch on their right leg, would that information get through? No, it's cut right here. But how about if we pinch their foot and we use pressure or temperature, put a cold plate down here? Will that information get through? It certainly will. But from the other side, from the left leg, I should probably put a T in here. From the left leg, what we see is fine touch and proprioception get through just fine. But pain and pressure and temperature from the left leg are now intercepted, and they're broken here because we've broken the wires at this point. So if you can walk your way through that, and you know what's going on with each of these cases, you understand the projections of these two pathways. And it's a worthwhile exercise to do. Spend some time with this, because this will illuminate how the wiring pattern is working out for the DCML, which, remember, this is going to be proprioception and fine touch, and the corticospinal tract, which is going to be pain, temperature, and pressure. Well, let's look at some slides from where we were yesterday and see if we can summarize what we talked about. Now, this first slide is one on the tendon tap reflex. And it's a nice image. I like this figure because it pretty much summarizes everything we talked about in one straightforward figure. So here's the patellar tendon. Here's the quadriceps muscle connected to a tendon to the patella to the tibia on the lower limb. When we whack this tendon, we stretch the quadriceps. When we stretch the quadriceps, we're now opening stretch-activated channels in the input region of this primary sensory afferent. that will produce a generator potential. And if we have enough stretch-activated channels opened, that will generate a generator potential large enough to reach threshold and fire action potentials that propagate along the conductor region of this primary sensory afferent all the way into the central nervous system. And in this case, the primary sensory afferent is going to release the neurotransmitter glutamate onto both this inhibitory interneuron and this green lower motor neuron. Note that the lower motor neuron innervates the quadriceps muscle. So that's the circuit. That's the circuit. That's why your foot jumps. That's why you move your foot when you pull on this sensory input region. When you do that now, the action potential arrives at the output region of the primary sensory afferent, releases neurotransmitter, sets up an EPSP, many of them probably, and that generates an action potential which propagates to the muscle, where in the muscle, that lower motor neuron releases acetylcholine and starts the process of muscle contraction. But at the same time, remember this primary sensory afferent is releasing glutamate, excitatory neurotransmitter, onto this red or orange inhibitory interneuron. So this excitatory neurotransmitter is going to turn on the inhibitory interneuron. It's going to generate these it's going to generate EPSPs at this site. It's going to generate EPSPs that generate action potentials that propagate to this point. And at this point, that inhibitory interneuron will be releasing inhibitory neurotransmitter. Could be GABA, and the spinal cord is probably glycine. That inhibitory neurotransmitter now opens chloride channels at 8. It opens chloride channels in this magenta lower motor neuron, which innervates the hamstrings. So what this means is when in this loop, when we activate the primary sensory afferent and turn and cause the muscle to contract, we simultaneously inhibit the motor neuron going to the antagonistic muscle. So that's that circuit. And that process is called reciprocal inhibition. This is just a simple figure showing the sequence of events, and this now boils it down to the electrical events. And what it's showing here is when we tug on this membrane with the stretch-activated channels, that produces a generator potential. The generator potential exceeds threshold at the trigger zone. We fire action potentials, or a series of action potentials, that propagate along the conductor region of the primary sensory afferent to its output region on the lower motor neuron, where it releases the neurotransmitter glutamate. The glutamate now opens these glutamate ligand-gated channels, produces an EPSP. The EPSP then generates an action potential of the trigger zone, propagates now down the conductal region of the axon to the muscle fiber, and in the muscle fiber it will release the neurotransmitter acetylcholine, causing muscle fiber contraction. And each of these steps now shows how we have these generator potentials and synaptic potentials and action potentials. And again, down here, we'll produce an EPSP in the muscle fiber and an action potential that causes muscle contraction. Then the next slide shows not so much the story of this reflex arc, but now we're beginning to think about how we're going to carry information into the brain. And that's shown by this figure. Here's the neuronal cell body. Here's the process that enters the spinal cord. Now, the one we just showed was how this process would go into the ventral horn, but a branch of this neuron is going to enter the dorsal column system. And it's going to ascend the cord on the same side as the site where it entered. And this is showing the conductal regions. This is showing the white matter. And so with the white matter, we see up here, these are the dorsal columns. The information that's going to be carried in this dorsal column is going to be information on primary sensory afferents that are conducting information about proprioception and fine touch from this side of the body. These dorsal columns are going to be also containing the axons or conductor regions of primary sensory afferents, carrying information about proprioception and fine touch from the left side of the body. Now, down here, this pathway is the so-called spinal thalamic tract. Now, what's going to happen here is the primary sensory afferent is going to come into the gray matter in the dorsal horn, synapse onto a second-order neuron. The second-order neurons are going to cross the midline, enter the spinal thalamic tract, and ascend the cord all the way to the thalamus. and synapse in the thalamic relay nuclei. Whereas the primary sensory afferents in the dorsal column system are going to synapse onto second-order neurons in the dorsal column nuclei. Now, this is a drawing. It's done by a colleague of mine a number of years ago, but it's a very simple drawing, but I really like it a lot because it shows the pathways. Now, this is going to be the dorsal column system or the posterior column system. And this is the primary sensory afferent. It has this neuronal cell body in the dorsal root ganglia. Sends a process in. It will form a synapse onto lower motor neurons down here, but also it sends a process into the dorsal columns that extends to this single neuron, extends all the way up to the dorsal column nuclei, where it synapses onto the second order neuron, which now sends its process in the medial lemniscus over to the third order neuron, which now synapses onto neurons in the thalamic relay nuclei, which go to the somatosensory cortex. Let me see if I can center that better. If that doesn't help. No. Okay, so the next figure shows, of course, the other pathway, the spinal thalamic tract. And again, here's the primary sensory afferent, enters the dorsal horn, synapses onto the second order neuron. Second order neuron crosses over the midline, enters the spinal thalamic tract, and ascends all the way up to the thalamic relay nucleus, where it synapses onto third order neurons that are now going to go and synapse onto neurons in the somatosensory cortex. So that's basically everything we talked about yesterday. And what I want to do now, though, is begin thinking about what's going on in the somatosensory cortex. And then we'll come back to that slide that was just shown there briefly and talk about its position within the brain itself. So over here on the right-hand side, what I want to talk about is the organization of the somatosensory cortex. Now, when we talk about the cerebrum, we can divide the cerebrum into these two major regions, one of which, of course, is going to be white matter. And much of the cerebrum of the brain is just this white matter, basically axons connecting one part of the cerebral cortex to the other. The other major part of the cerebrum, though, is the cerebral cortex. The cerebral cortex is gray matter. It's neuronal input regions. And if we look at the structure of the cortex, wherever we look, now we're going to look at the somatosensory cortex here, but wherever we look, turns out there are 1, 2, 3, 4, 5, 6. There's 6 layers of neuronal cell bodies. Don't remember the number, but it's just true for wherever we look in the somatosensory cortex, we have 6 layers of neuronal cell bodies. When we're talking about the somatosensory cortex, and we're talking about the third order neurons originating in the thalamic relay nuclei, these send processes into the somatosensory cortex, and they synapse onto neurons in layer 4. Again, that's a detail. You don't need to know that, but it gives you some sense of the fact that we're consistently using these third order neurons to synapse onto a particular set of neurons in the somatosensory cortex. It's the pattern. And we can do this on both sides. If we look at the other side, again, we have the thalamic relay nucleus. We have a neuronal cell body of the thalamic relay. The third order neuron sends a process up. And now innervates, again, same pattern. Let me get this out of here and then put the layers in. 1, 2, 3, 4, 5, and 6. And third order neuron will go to layer 4. That's true for all the third order neurons. So there's this pattern here. We don't quite understand all the synaptic integration that's going on among these 6 layers. That's something that's under rigorous investigation right now. So a lot of interaction among these. But we do know that these third order neurons go to neurons in the somatosensory cortex in layer 6. I'm sorry, in layer 4. The other thing we know is that in the somatosensory cortex, there's something known as somatotopy. And somatotopy is defined as a map of the body plan. On the neurons in the cerebral cortex. And what it means is that if we would follow the neurons coming from a particular part of the body, that there will be a region in the somatosensory cortex where there'll be neurons from the foot. And there'll be a region where there's going to be neurons innervating the head. And regions from, I think it actually should be hand here, and regions from the head. And I'll show you an image of this in a bit, but what it means is that we have a disproportionate innervation of the somatosensory cortex depending on the number of sensory neurons, third order neurons arriving at those sites. And if you look at different animals, you get different patterns. If you would look at the somatosensory organization of an elephant, there'd be an enormous part of its somatosensory cortex that was dedicated to its trunk, because that's the way an elephant interacts with the world. It interacts with the world through its trunk. If we were to do this on a horse, you'd find that much of the somatosensory cortex is dedicated to the lips of a horse, because again, horses don't feel their way through the world. They basically use their lips to get through the world. Now, if you look at human beings, because of the way we have an enormous part of the somatosensory cortex dedicated to our hands, because so we do by our hands. After all, a dog or a cat can't play the violin, but some people can play the violin, and that's because they can use their hands, this tiny fractionated movement. So an enormous amount of sensory information comes in from the hands. We'll see an enormous amount of sensory information also comes in from the face. Because as we speak, as we make language, as we use our lips and our tongue and our jaws to make language, we need to know where they are in space. We need the proprioception. And by having that proprioception, then, we can know where they are in space, and we can use that information to generate, through the motor cortex, movement. But what we'll see here is that there's a disproportionate amount of the somatosensory cortex that's taken up depending upon the particular species and how they use their somatosensory system. Well, let's look at a couple of pictures to try to get things a little bit better organized in terms of the brain. So what we're looking at here is a simple view of the cerebrum. And it's as you're looking at the left side of the cerebrum. It's as though I'm standing here and facing that wall. Now, we could do this to anyone in this room. We could basically take their skull and pop off their skull, and we would see this pattern. We would see there's a region of the brain up here, which is under the frontal bone. This is called the frontal lobe, or we'll see here in a moment, the frontal cortex. Behind that is a region called the parietal lobe. The parietal lobe is that region immediately behind the frontal lobe. And we're going to see here in a moment there's a separation between the frontal lobe and the parietal lobe in a particular region, this infolding here called the central sulcus. One of the things you can see from this figure is that when we think about the cerebrum, in order to get all of those cells packed into the brain case, In order to get all the cells in there, we throw the cerebrum into these folds. And that's what we're looking at here, are all of these folds. And basically, if we could take anyone in this room, we would see the same pattern of folds. And we would see that there's a region here called the precentral gyrus. It's called the precentral gyrus because this gyrus is a kind of plateau. So fold, we climb to the top of the fold, we come over the top of the fold, that's the top of the plateau, that's going to be this gyrus. But the gyrus then also will dive down, it will continue, and the space between these adjacent gyri will be called the sulci or sulcus or fissure. So what we're seeing, and I'll draw this in a minute, is the way those folds are organized is they have a top, sort of like the top of my hand, but they also have this region up this side and over the top. And that, the top of that's going to be a gyrus. And then there'll be another one. I don't have 4 hands, but see there's another one over here, and those 2 gyri will be separated by a sulcus, a fissure, a space between the 2. And that's what they're trying to show in this figure. They're trying to show that there's this space between the precentral gyrus and the postcentral gyrus. And the precentral gyrus here is shown in pink. Now what they've done is they've taken this little slice out, and they're trying to show what the cortex would look like. And as we mentioned just a minute ago, when we talk about the cerebrum, we have both white matter and gray matter. The gray matter is found as the cortex. The gray matter are the 6 neuronal cell body layers in that very thin veneer, no more than a couple of millimeters thick. And that's shown here. So this is the cortex. But beneath that, shown sort of in this sort of flesh-colored structure, that's intended to show white matter. That's intended to be the axons which have been ensheathed by the myelin. And so that's going to be white matter. Most of the cerebrum is going to be white matter. Most of it is connections between different parts of the brain. But the synaptic integration part is going to occur out here in the cortex. And that's what we drew in the board. What we drew in the board is this region here, the somatosensory cortex, which is the cortex which is in this structure. So if we'd open this up, we'd see the 6 neuronal cell body layers as part of the post-central gyrus. So the next slide, I should point a couple other things out here because we're talking about the brain, just what they're doing. This is the frontal lobe. Much of your executive function is found in the frontal lobe. There's also some motor stuff found in the frontal lobe. We'll see that this is the motor cortex. There's some regions just in front of that which are called the premotor areas, and there where planning for motor activity happens to occur. And then we'll send that information to the motor cortex, and we'll see here later today that the upper motor neurons in here will send information to lower motor neurons to tell them to become active. The parietal cortex we see here in the postcentral gyrus will be the somatosensory cortex. But the parietal cortex does a whole lot of other stuff too. The parietal cortex, we have what is known as the association areas. Association areas are where we have all manner of different kinds of sensory input. Some of it visual, some of it auditory, some of it taste and smell. All of that comes into a region in the parietal cortex where that information is integrated. That's how we figure out what reality seems to be around us, by interpreting all of that sensory input at one time, and from that generating what we believe to be the reality that we know. That's all done largely in the parietal cortex. This region back here, the occipital cortex, is largely dedicated to vision. The temporal cortex does a whole bunch of things, but in part it's important in language and language expression. So what we've done so far, though, is we've gotten the information to the somatosensory cortex. And this next figure is to show the somatotopy in the somatosensory cortex. So again, it's this, if you take that upper right-hand bit, it's as though I'm facing the wall. And what we're looking at in blue is the postcentral gyrus. In red is the precentral gyrus. And now what we've done is we've opened up this region of the postcentral gyrus, and we're looking at just the cortex. And we're looking at the neurons in the cortex that are involved with different parts of the body. And you notice there's this enormous region that's dedicated to the face and the jaw and the tongue. That's because we need to know where our faces are in space when we speak, along with our tongue and our jaw. We have an enormous area dedicated to the hands here, much less to the head itself and to the trunk and the legs. So when you extend that out into a three-dimensional structure, you get something like this. This is a three-dimensional homunculus. And notice what it's trying to demonstrate, that there's a disproportionate area that's dedicated to different parts of the body. For human beings, so much of it is dedicated to the hands. But there's an enormous amount dedicated to the lips and the tongue. I mean, Mick Jagger doesn't have lips like that. That joke's aging with both me and Mick Jagger. But this is an exaggeration of the somatosensory cortex, showing that we have this enormous area dedicated to hands, not much to feet or legs, and a large amount to face. If you do this with a rat, you'd see tremendous amount of information coming from the vibrissae, the whiskers, because the whiskers are the way rats interact with one another and their environment when they're crawling through dark spaces. So depending on the particular animal and how it's evolved, we'll have different somatosensory systems, which will then be emphasized in one way or another. We emphasize the head and the hands, whereas other animals will emphasize things like their whiskers or their lips. But that's the story of the somatosensory cortex. And I don't have time to talk about it, but the information in the somatosensory cortex is going to go out to other parts of the brain. It's going to go out to other parts of the brain, particularly the parietal association areas. And then in the parietal association areas, the image of the world will be made. And then in the parietal association areas, information will be sent forward to the forebrain, basically sent forward to the forebrain, where we have executive function. We'll decide what to do. That executive function will make decisions and send information then to the next area called the motor cortex. And that's where I want to go now and talk about the motor cortex and descending pathways. So let's take this panel and describe it as the motor cortex. And descending pathways. So again, we can imagine the motor cortex, much like we saw the somatosensory cortex above. Now, the motor cortex is going to be located on a place called the pre-central gyrus. It too will have the same plan as we saw before in the somatosensory cortex, where there'll be 6 neuronal cell body layers. In layer 5, there's a set of special neurons. In layer 5, there's special neurons called upper motor neurons. We abbreviate this as the UPM. They also have a name called Bet cells. Betz was a 19th century Ukrainian physiologist, and he discovered these things, so we were stuck with the name. These are gigantic cells. They're like 100 micrometers in diameter, and these neurons send a process out that enters a region in the brain called the internal capsule. And the internal capsule is a bundle of axons. And for our purposes today, these are going to be a bundle of axons of upper motor neurons. So these red Bet cells will enter this region called the internal capsule. They'll skirt around the thalamus. And they will enter a region of the brainstem called the medulla. And just before this brainstem forms the spinal cord. These upper motor neurons, I'm just showing one, but there'll be literally thousands and thousands of these, crosses over the midline. So again, here's the midline. And these neurons cross the midline in an area called the decussation of the pyramids. The decussation of the pyramids gets its name because the early anatomists, when they started sectioning through here and looking at these pathways, it looked like a triangle. But then when they started to reassemble it in 3 dimensions, it looked like a pyramid. So it's called the decussation of the pyramids. These upper motor neurons then enter part of the spinal cord. This is a region of the cord called the lateral corticospinal tract. Now, we can dissect this terminology, tract. We know that's going to be a bundle of conductor regions in the central nervous system, so it's a tract. It's lateral, it's going to be on the side of the spinal cord, but the more important terminology here is corticospinal. It tells us the neuronal cell body is found in the cortex. It tells us this output region is in the spinal cord. So let's draw one of these spinal cords. Let's imagine that we take a section through here and look at the spinal cord in cross-section. Again, this is the gray matter. There's our central canal. We can imagine in red over here is the lateral corticospinal tract. Now this is chock full of conductor regions, axons, whose cell bodies are on the opposite side of the body because they originated on the opposite side in the somatosensory cortex. These axons, and I'll just put these as dots, during development, they will leave the corticospinal tract and then at just the right spot, they will leave and enter the ventral horn. So this is the ventral horn. Remember, when you see the term horn, we're talking about the central nervous system. We're talking about the gray matter. So this upper motor neuron axon, whose cell body resides in layer 5 of the motor cortex, that individual, this great long axon now synapses here onto a lower motor neuron. And as we've seen several times now, this lower motor neuron is going to exit the cord. It's going to enter in the periphery the anterior or ventral root and find its way to a muscle where it's going to innervate muscle. This is innervation of muscle by the lower motor neuron. The lower motor neuron now is activated by the upper motor neuron. So we have a single synaptic pathway. The upper motor neuron extends all the way from your motor cortex down to your brain, into your brainstem, crosses over the midline in the medulla, enters the spine, finds its way to the appropriate site, where it's now going to innervate the lower motor neuron. So any questions on that? Yes?
So just to clarify, brain pathways don't need the second order neuron to cross the midline?
Say that again, please.
So just to clarify, for the descending pathways, it doesn't need that second order neuron to cross the midline. It's just one continuous
So let's keep separate. This is a question about the descending pathway. Let's keep separate the somatosensory system and the motor system. The somatosensory system, the afferents, the information coming in, that information goes through a 3-cell pathway. The information going out, at least in this particular system, in our system, goes through a 2-cell pathway from upper motor neuron directly to a lower motor neuron. So we don't normally consider those, at least as far as I know, we don't use the nomenclature primary, secondary, and tertiary, or second or third order for the motor. We simply talk about an upper motor neuron synapsing onto a lower motor neuron. This is not the question.
Is the anterior/ventral root, is that arrow pointing at the exusion, I suppose, of the neurons coming out?
What I'm trying to say, the question is, what am I talking about here? Remember that there's a kind of transition between the central nervous system and the peripheral nervous system. So I'm trying to get that idea across. Trying to get the idea across that the lower motor neuron has its cell body in the ventral horn, which is in the gray matter of the central nervous system. It now will send a process that goes out into the periphery. This is a somatic efferent. It's going to innervate muscle. So in order to find its way to muscle during development, it sends a process out into the ventral root. So now you're in the peripheral nervous system. And then from that ventral root, you'll actually someplace up here fuse with that information going to the sensory side. And that's now going to form the spinal nerve. Okay? Good. Yes?
What is CNM or LMN stand for?
Lower motor neuron, LMN, and UPM, upper motor neuron, UMP, UMN. In a minute. Let me try one last thing. Let me lay this out, and then we'll come back to it. We'll come back on Monday. What I'm going to do is lay out a series of muscle fibers. So these are muscle fibers. And I want to emphasize the idea that we have a lower motor neuron in the spinal cord. This lower motor neuron will send its process we showed above out to this, and it's going to innervate muscle fibers. But each lower motor neuron innervates several muscle fibers. But each muscle fiber receives input from only one lower motor neuron. And that will set the idea for Monday of talking about the motor unit. The motor unit is the lower motor neuron, group of muscle fibers it innervates.